Good morning, good morning, good afternoon, or even a late good evening, wherever you are around the world. This is the International Space Science Institute, again with the Game Changers seminar series, resuming after the end of the year holiday season last year. Following a discussion in the directorate, we felt we should pick up on a subject that is often discussed and used these days in solar system and exoplanet science: habitability, defined as the capability of an environment to sustain life and loosely linked to the stability of water on or close to the surface. But we all know it's much more complicated. There are conditions of the survival of life defined by the galactic environment, as well as there are microbial life sustaining a habitable microenvironment. And then there is an interaction, at least postulated interaction, between the environment and life on a planetary scale. And we are, as human beings, even doing our own experiments, with the habitability of our own planet. These subjects and more, we will be dealing with in this series of 10 talks that are loosely organized along decreasing scales, so from galactic to microbial. However, because scheduling is a challenge, the talk foreseen for next week on galactic habitability needed to be moved to the very end of the series. So next week we will not have a seminar, but then the week thereafter. But we felt that the series should be started off with a presentation and discussion on the origin of life to sort of prepare the stage. And we feel very and extremely fortunate to have been able to recruit Antonio Lazcano, the author of the very entrance in the Encyclopedia of Astrobiology, to give this talk. By the way, if you want to stay abreast of the seminar and other ISI activities, consider subscribing to our newsletter. You can do so at the bottom of any of the ISI web pages. Now, Antonio is a professor at the Faculty of Sciences of the National Autonomous University of Mexico, where he specializes in evolutionary biology. He is one of the world-leading experts in the subject, having published extensively on the subject. He served as the president of the International Society for the Study of the Origin of Life for 2 terms, and as honorary director of the Lynn Margulis Center for Evolutionary Biology in the Galapagos Islands in Ecuador. He has worked at many institutions around the world and received various awards, among them the National Researcher Level 3 Award as National Science Council of Mexico and the Medal Guillaume Medal of the Collège de France, among other distinctions, and he is a member of El Colegio Nacional de Mexico. For the question of answer after Antonio's talk, we invite you to use either the chat channel, in which case I will read your question to Antonio and all the other participants, or you raise your hand and then Willie will give you the floor and you can speak for yourself. With that, I stop, and Antonio, the floor is yours. We're looking very much forward to your presentation.
Thank you so much, Professor Spahn. Thank you so much, Tillman. I have been lucky enough to be in the International Space Science Institute several times. And I must say that I'm very, very fond of Bern and the academic atmosphere that you have there. And I want to thank you for the chance to talk to you today about the origins of life. And since this talk is hosted by the International Space Science Institute, I thought that I might as well start with trying to describe a little bit what was the astrophysical context of the emergence of life on Earth. And needless to say, life on Earth is the only example that we have of biosphere in the universe that we know about. But there are many unknowns about the origins of life. One has to say actually that we really do not know in detail how life appeared. on Earth. But we know that life is a very ancient phenomenon on Earth. The oldest fossil evidence that we have for living systems in our planet is about 3.4 to 10 billion years ago. And the Earth, we know that was formed about 4.6 billion years ago. And that places the origin of life, whatever the way it took place, quite close to the formation of the Earth. Now, when one thinks of the conditions of the early Earth, one must realize that we had a very active sun at the time. It had a higher UV flux than today. It had a stronger solar wind that we see today. It was a very active star. And clearly that must have shaped in many, many different ways the evolution of the origin and the early evolution of life on Earth. Now, everybody's familiar with this scheme that I included here about the formation of the Earth, the idea that was put forward by Immanuel Kant, the philosopher. And in fact, the idea that the solar system formed by the condensation of an interstellar cloud is strongly supported by the fact that we do see that phenomenon ongoing right now. We see, for instance, in the Orion Nebula, a huge number of planetary disks, of protoplanetary disks, places where in fact this condensation, this collapse of interstellar clouds are taking place, leading to early stars and the formation of protoplanetary disks among them. And one of the most fascinating issues about radio astronomers when it comes to the exploration of this kind of system is the fact that you see a huge number of interstellar molecules. And that in itself is fascinating, but from the viewpoint of the origins of life, what makes this extremely attractive is the fact that, first of all, most of the molecules that we see in the interstellar clouds are actually organic molecules, namely, they have at least one atom of carbon and covalent bonds, which is our definition of an organic compound. And secondly, some of the molecules that we see in these clouds where planets are formed are molecules like hydrogen, water, ammonia, formaldehyde, hydrogen cyanide, methane, and so on, which are molecules that we believe were actually part of the primitive environment that led to the formation of organic molecules in the primitive Earth. And this immediately raises the issue of what, if any, role these molecules, which must have been present in the solar nebula, what role did they play in the actual origins of life? The other fascinating issue that we have to take into account is that perhaps one of the most surprising results, and people are normally not aware of this, but one of the most fascinating results of a space exploration in our own solar system is the realization that the early solar system was actually shaped by an extensive period of collisions. We see very few Precambrian craters in the Earth itself, because the Earth is a very active geologically planet. But if we look at the moon, as you can see in this wonderful photograph taken in the moon itself, you can see the huge number of collisional craters that are indication of this very intense bombardment that the moon suffered in its early history. The moon being so close to the Earth, it's very reasonable to assume that an equivalent process took place on the early Earth. Now, the astronomers, the geologists, the planetologists can actually classify the kind of meteorites that are able to form these craters. They are constantly falling on the Earth nowadays. And we know that we can broadly divide meteorites in three great classes. The stony meteorites that look like a normal stone, the ones you will see in the field, for instance. And these represent a huge number of this huge percentage of meteorites. We have the stony iron meteorites, which are quite rare, and we have the iron meteorites, which are extremely easy to identify identify once you see them. And if we concentrate on the stony meteorites, we can divide them in two broad classes: the chondrites, which have an inclusions of silicates and carbonates, and the achondritic meteorites. But the chondritic meteorites have a subclass, the carbonaceous chondrites, which are extremely rich on inorganic compounds. And we were quite lucky in 1969 that we were able to witness the fall of one of these stony meteorites, the so-called Murchison meteorite that fell in Australia. Australians are very organized people, and very rapidly they connected. It fell in the field close to the town of Murchison. They very rapidly collected the pieces of the meteorite, and when you break one of these meteorites, one of the pieces of this meteorite, you can see that it really looks like a stone, and you can see these inclusions that I have already mentioned. There's one point that I would like to emphasize in this picture, which was given to me by my friend David Immer at the University of California, and it's the fact that you see a crust in the meteorite, like if it had burned, like a burnt cake, like a burnt piece of bread. And that's exactly what happened. When the meteorite was falling across the atmosphere, the terrestrial atmosphere, it got heated. The heat altered the outer part of the meteorite, but the stones are very bad transmissions of heat. So the effect of the heat was limited to the outer part of the meteorite, and then we can analyze the meteorite without any problem. Why do I say that we were quite lucky to see this meteorite falling in 1969? Because by then NASA had already set up the laboratories to analyze the moon samples, and we were thus able to analyze this meteorite without the risk of contamination. And the results were absolutely fascinating. Once again, this picture was given to me by my friend Dave Dimmer, and you can see that the meteorite has a huge percentage of what we call kerogen, which is a very complex organic polymer like the asphalt, somehow similar to the asphalt that we use in streets, but there is a significant portion of soluble organics, and when we analyze them, we find very much to our delight that in the 4.6 10 to the 9 years old meteorite, we see a number of organic molecules like decarboxylic acids, like amino acids, like amides, like purines. They are molecules that we see in living systems. Now, we don't have a sample of the primitive Earth. We don't have a sample of a piece of stone of the sediments when our Earth on Earth was formed, but the meteorites are sort of geochemical fossils of the events that were taking place when our own planet was forming, was condensing. So what these analyses are telling us is that there were a number of organic chemical syntheses that were taking place in the solar system that led precisely to the kind of organic compounds that we see nowadays in living systems. And this is fascinating. So indirectly, we can say that because of the analysis of the interstellar clouds, because of the presence of organic compounds in comets, I have not spoken about them, but you know what they are, and because of the analysis of these kind of meteorites, we have a clear demonstration of the synthesis, of the non-biological synthesis of organic compounds. Now, for many, many years, we were truly embriagated by the results coming from the Murchison meteorite, and I have used the word quite on purpose because we do see alcohols in the Murchison meteorite. But the recent work of the people from NASA, led by Jason Dworkin, by Danny Glavin, and other friends and colleagues in the United States, have demonstrated that, in fact, there is a huge array of amino acids that we can discover in the different meteorites. You can see, of course, the very simple, simplest amino acid, which is glycine, but you see also a huge number of amino acids. And in some cases, if you're looking at this table, which is a little bit overwhelming, but you see that we actually can detect and find for instance, a number of amino acids with 6 carbons that we cannot identify because we do see the signals during the chemical analysis of the meteorite, but we don't have the standards to compare to them. So this is telling us that the chemistry leading to the formation of amino acids in the primitive solar system produced a huge array of organic compounds, including precisely the ones that we see in proteins. Now, in addition to amino acids, we actually can see without any problem in different meteorites, the ones indicated here in the X axis, we can see the presence of different amounts of nucleobases, the same bases that we see in RNA and DNA. So what this is telling us, this graph is telling us, is that really one should undergo a more detailed analysis of different kinds of meteorites to actually be able to produce a sort of taxonomy of them and the kind of organic compounds they have. But the presence of nucleobases, one in adenine, epoxantic, xanthine, and so on, is very clear. And it demonstrates that you can also have the components of nucleic acids formed by these kind of reactions in the primitive solar system. And I think we can summarize what I'm just saying about the nucleobases by showing you this wonderful table that was published about 8 years ago or so, in which you see all the compounds that we can detect in meteorites, all the ones in black, and in the prebiotic simulations of the primitive Earth. Now, the presence of these organic compounds in samples from outer space actually gives a strong support to the proposal made in 1924 first and in 1936 by Alexander Ivanovich Oparin, biochemist, a Soviet biochemist, that proposed that in fact the first living beings, the first living entities were bacteria that were unable to synthesize their own food. They were nourishing themselves from the organic compounds present in the so-called primitive soup. And these were formed in the primitive Earth because the Earth had no oxygen in the atmosphere. In other words, by doing a superb compilation of information coming from different fields, astronomy, geology, chemistry, biology, physical chemistry, and so on, Oparin was able to propose this stepwise scheme of the origins of life by assuming that the primitive Earth had no free oxygen. And I will return to this a little bit later on. But the basic point is that now we have evidence that, in fact, you can form organic compounds not only before the origin of life took place, but actually before the origin of the planet. And this book was precisely the basis of a very famous experiment that I'm sure you are all aware of, the experiment published in 1953 by Stanley Miller, very young chemist and a graduate student of Miller-Urey, one of the founders of modern cosmochemistry. And after much debate with his advisor, Stanley Miller got permission to work for 6 months on a simulation of the primitive conditions of the Earth, using as basis, as I already mentioned, the Oparin hypothesis. And so he built up a very small apparatus, and in the upper flask of this apparatus, he put methane, ammonia, hydrogen, and water vapor that was coming from the lower flask, and the vapor mixed with all the other components of this simulated primitive atmosphere. It was subject for a week to the action of electric discharges. And in only a few hours, you can actually see how the organic compounds are formed. You can see the actual formation of a polymer that we can then analyze. And now many of us ask many times Stanley Miller what happened with the original apparatus. And he said, well, I put it in the trash bin because it was very complex to clean up. But our good friend Bill Schoff made repeated the experiment in his lab at the University of California in Los Angeles. And you can see how indeed in very few hours, you can see the synthesis of organic compounds taking place. Now, I always like to take this photograph, I always like to show this slide, because you can see a photograph of a very elegant Stanley Miller. Of course, the photograph was kept by his mother. And you can see how the people used to go in the lab at those times, white shirt, tie, cufflinks, and so on. And believe me, the situation has changed a little bit when I look at my own students in my lab. But the fact is that the experiment was extremely successful. Stanley Miller did paper chromatography, which was the range of chemical analysis at the time. And he was able to publish this paper chromatograph, in which you can see the presence of aspartic acid, glycine, alpha-alanine, and a number of additional amino acids. We were lucky enough to have the samples of the original experiment, actually samples from all the experiments that Stanley Miller did in his 50 years of professional life. And at the lab of my good friend Jeff Beda, now at UC San Diego, we compare the organic compounds, a few of them actually, aliphatic amino acids that were formed in a simulated experiment like the Miller one, compare them with the presence of amino acids in the Murchison meteorite, and you can see how there is an excellent correlation between the results that we have in the experiment with the amino acids present in the meteorite. So this is telling us that our simulations of the primitive Earth are worth. I mean, we have a control, we have a comparison with the samples coming from outer space, and actually in terms of quantitative and qualitative comparisons, we can actually see that our experiments actually simulate what we see in small bodies from that have the age of the solar system. And of course, it has lent a strong support to the idea that there was indeed a primitive soup, and I would like to say that one shouldn't think of the primitive Earth as being homogeneous. I think that it's fair to say that it was a very rich environment in which you could have volcanic activity, hydrothermal vents, the infall of comets and meteorites bringing in, of course, organic compounds. You could have electric storms. You can have microenvironments like ponds near the sea. And probably the Earth at the time had no continents as we think of them today, but rather was spectred all along the planet by a number of systems of small volcanic islands. Needless to say, there is a debate about which is the best place to synthesize organic compounds. But all in all, I will say that our different simulations of these kind of environments lend support to the idea that we had a primitive soup with amino acids, nucleobases, sugar, lipids, biochemical oligomers, and many kinds of catalysts like metallic ions, like clays, and like some amino acids like proline, for instance, that can be very, very efficient catalysts. Sometimes when I look at the literature of prebiotic synthesis, I think that too many cooks have spoiled the soup, as the British like to say. But the fact is that, in fact, we do see that under different conditions, we can synthesize the organic compounds. Now, one thing that is frequently forgotten is that when Stanley Miller published his paper in 1953, he had been preceded by 3 weeks by the publication of the Watson and Crick double helix model of DNA. And this truly changed all of biology, not necessarily for good, because I must say that for several decades, biology became strictly concentrated on the molecular, the subcellular structure of life, and the naturalists, like the paleontologists, the physiologists, and so on, suffer for a while because they were displaced from the focus of attention of the funding agencies, of the students, of publications, and so on. We are coming to a sort of equilibrium. But the fact that the harvest of 1953 included In the context of the double helix model, the Miller experiment clearly indicated that you cannot have a theory on the origins of life that is not consistent with what we know at a molecular level of life, of cells. And I think it's fair to say that after the 1953 Miller-Urey experiments, attempts to understand the origins of life were shaped very, very strongly by the emphasis in unraveling for trying to understand the details of DNA replication and protein biosynthesis. And something that I have to underline, being in the International Space Science Institute, by the development of the space programs. Without NASA, we would not have this extraordinary development of the origin of life field. Without the space programs, the European space program, the Chinese, the Japanese, the American, the Russian space program, we really would have not gained the extraordinary insights that we have today about cosmochemistry, chemistry of in other parts of the universe, about the presence of planets, and of course about the evolution of the solar system. But in 1953, what Stanley Miller has synthesized were amino acids, and the people were wondering, what about the other components of living cells? And the solution came in 1960 when Juan Oro, a Catalonian chemist living in the United States, was able to demonstrate that the condensation of hydrogen cyanide, which we have already seen is quite abundant in the interstellar medium, that is readily formed in the Miller type experiment, that we can see in cometary nuclei. Hydrogen cyanide condenses very easily to form a dimer. Then there is another condensation reaction that gives a trimer, another one that gives a tetramer, and then with some components that John was able to demonstrate were formed by the hydrolysis of hydrogen cyanide, you see amino imidazole carbonitrile, which is a very reactive molecule, I have to say, and a condensation with formamidine gives adenine, which is of course one of the components of RNA and DNA, and of course it's present in ATP. And in other words, you can form under primitive air conditions without any problem. You can easily form one of the main components of genetics and metabolic bioenergetic systems on Earth. Later on, Jim Ferris and Leslie Orgel were able to demonstrate that if you have a water solution of the amino malonitrile and put it in the sun, they went actually out to the beach in the Salk Institute, it easily undergoes photoisomerization, then the formation of the amino imidazole carbonitrile, and then adenine. In other words, you only need water, sunlight, and hydrogen cyanide to be able to form one of the main bases, nucleobases that we see on living Earth. And this, of course, immediately raised the connection between molecular biology and the study of the origins of life. Now, the presence of the bases of a prebiotic synthesis of the bases was extremely encouraging for a number of scientists, including those that in 1950 had formed the so-called RNA tie club, an idea that was proposed by George Gamow, the very, very distinguished physicist who wrote beautiful books, popular science books, and James Watson. And they came out with the idea of forming the so-called RNA tie club that was created in the Cosmos Club in Washington. With the word goes around that it was formed there because the whiskey was very good and quite inexpensive. But when you see the list of the people forming the RNA tie club, you are taken back by the name of the scientists: Alex Rich, George Gamow, Edward Taylor, Charles Gaff, Stent, Watson, Orgel, Delbruck, Crick, Benner, Calvin, Feynman. The number of very, very distinguished scientists is quite surprising. And what is missing, of course, is not only a number of scientists here, but also female colleagues. One cannot rewrite the past, but one has to understand the conditions there were at the time, in which unfortunately very few female colleagues were part of this group. And the situation, believe me, has changed for the good. But the important thing is that people were paying attention to RNA. extremely attention. And then in a few years later, viruses were demonstrated to have, some of them, RNA as genetic material. In fact, one of the co-authors of the paper that led to the demonstration of the presence of RNA genomes in viruses was none other than John D. Bernal, the extraordinary British scientist who had been the advisor to Rosalind Franklin, to Max Perutz, to Francis Crick himself. And don't forget that many people believe that since viruses are very small, are very simple, and can be crystallized, a number of people still think that they may be at the threshold of life. So many people think that they are primitive, which is not really the case. So since viruses were in this threshold, at this undefined twilight zone in the eyes of many between the living and the non-living, and many thought they were primitive, and some of them have RNA genomes. A good example is the SARS-CoV-2 virus. Many people believe, okay, then RNA genomes must be primitive. In other words, as the English say, a broken watch gives the right time now and then. There was a wrong premise, but the right conclusion. And so the idea began to spread in the scientific community that RNA came before DNA as genetic material in the course of evolution. And this idea was reinforced when the biochemists realized that okay, proteins are essential constituents of living systems. Proteins do most of the catalysis that we see in biological systems, but most proteins require coenzymes, the vitamins. Most of them require coenzymes, and quite independently, a number of scientists, Philip Handler, Arthur Yakin, Leslie Orgel, Harold White, realized that many coenzymes are actually derivatives of ribonucleotides, the components of RNA. And they proposed independently that perhaps this was indicated that in a very early stage of biological evolution, we had no proteins, but perhaps coenzymes, ribonucleotide coenzymes, were actually the ones driving an early an early stage of metabolism. And then in the 1960s came the extraordinary realization that RNA is extremely complex. RNA can plays a key role in protein biosynthesis. You need a messenger RNA, you need the RNA in the ribosome, you need the transfer RNA, which carries the amino acids into the ribosome. And then the people say, okay, we can have protein synthesis without DNA if we have, let us say, an RNA viruses. And then what for me is an extraordinary, truly extraordinary example of biological intuition spiced by an evolutionary perspective was a realization, independent realization by Leslie Orgel and Francis Crick, that if you look at DNA, DNA is a fascinating molecule, but it's basically inert in cells. DNA just has a very simple shape. Of course, the shape helps to protect the sequence of bases inside it from the hydrolytic reaction, from the water outside the DNA, double helix DNA molecule. But if you look at proteins like this hemoglobin protein, it's clear that the shape of the molecule, the three-dimensional shape of proteins, plays a key role in its biochemical interactions. And then, as I mentioned, quite independently, Leslie Orgel and Francis Crick predicted that given the extraordinary three-dimensional structure of many RNA molecules, RNA could play a catalytic role. And independently, I mean, their offices were side by side, but they decided to publish, to discuss these issues in different papers. They came to the idea that perhaps there was an RNA world prior to the present cells that we see today. In other words, that RNA existed not only prior to DNA, but also prior to proteins. And they proposed quite independently that in the primitive soup, in the primitive earth, there appeared an RNA molecule that could replicate, that was encoding for a catalytic RNA that was actually assisting in the replication of RNA. In simple terms, they predicted the existence of catalytic activity of RNA. Which is an extraordinary prediction. It came from two very distinguished scientists. The reputation of Francis Crick was quite extraordinary, but they predicted it and nobody paid any attention to them. Nobody paid any attention to them until 15 years later. It was demonstrated quite independently by the group of Sidney Ullman and Tom Scheck that in fact RNA molecules like those present in an enzyme that we call RNase, pRNase, which actually uses as a substrate transfer RNA to cut a piece of RNA. Sidney Ullman and his group demonstrated that this is the RNA component of this enzyme, the one that's catalyzing this reaction. And the group of Tom Scheck and his lab demonstrated that in fact RNA molecules by themselves can fall up by themselves in the presence of guanosine, a very simple monomer of a ribonucleotide, can actually cut a covalent bond, a phosphodiester bond, can form another bond, and RNA indeed is a catalytic molecule. And this was quite an extraordinary discovery. I was lucky enough to be a good friend of Leslie Orgel, through him to meet and speak with Francis Crick several times. And all of you remember that famous movie by Audrey Hepburn, Breakfast in Tiffany. Well, we normally had breakfast in Piatti, a lovely restaurant in La Jolla. And here you see the two of them, Leslie Orgel and Francis Crick. And once I was visiting La Jolla to work on a paper with Leslie Orgel, we went for a lunch, and I asked them, what do you think about your proposal has been proven that RNA has catalytic activity, that they are ribozymes, not only enzymes, but also ribozymes. And Leslie Orgel told me, well, if I had imagined that RNA had catalytic activity, in fact, and that persisted, I would have changed all my life and devoted myself to studying its properties. And Francis Crick, with a tongue in his cheek, said to me, no, I had already forgotten that I had made the proposal. Of course, he had not forgotten it. The problem was that, don't forget, the scientific environment, scientific milieu can be extremely conservative. And we have all been raised, educated, and unfortunately still tell the students that the main catalyst in the in biology are proteins, and we really paid no attention for many years about the possibility predicted by these two extraordinary scientists. So if we look at the RNA world hypothesis, I think it's fair to say that it emerged from the origin of life group, that the RNA world hypothesis has a long history, begins with the extraordinary work of chemists in the 19th century. They were able to synthesize urea, alanine, sugars, glycine, but they were thinking in terms of trying to understand the process of photosynthesis. They were trying to come out with models of the chemical reactions that take place in plants. They were not thinking about the origins of life. But all these results, together with others, were taken mostly by Oparin, also by Haldane, Lippmann, and Harvey, but mostly by Oparin, to come out with very detailed proposals of a heterotrophic origins of life. The Miller experiment and the synthesis of adenine gave a strong support to this idea of a primitive soup rich in organic compounds, from which the first living entities evolved. And then from here we go into the RNA world hypothesis. Now, we actually have very good examples of the key role of RNA in early evolution, but these are not limited to its properties as a genetic polymer and as a catalyst, but also by its many roles in extant biological systems. Perhaps the most impressive one is the fact that the ribosome is a ribosome. As Tom Scheck put it once in a beautiful essay published a number of years ago, the ribosome is a key component of all extant living entities. In the ribosome, we have the synthesis of the peptide bond. We have the synthesis of amino acids, of proteins. And as you can see in this lovely three-dimensional representation of the crystals of the small subunit and the large subunit of the ribosome that come from the group of Atlanta, Lauren William and a number of colleagues there, you can see how actually in the place where the synthesis of the peptide bond is taking place, we see no proteins. Without any doubt, we can say that is a ribosomal RNA, the one that catalyzes the synthesis of the peptide bond. So this is telling us that protein synthesis first evolved in an RNA system. But in addition to this key role in the synthesis of proteins, actually RNA plays many, many different roles in extant biological systems. If you want to have RNA, you need to have its monomers, ribonucleotides, the synthesis of ribonucleotides, the biosynthesis of ribonucleotides, the metabolism is absolutely universal. That means it's a very ancient metabolisms and probably was present in the ancestor of contemporary life forms. And once you have RNA, you can have coding RNA, like let us say in the SARS-CoV-2 virus, where you have the RNA encoding for the proteins that the virus can make, or the messenger RNA in our cells, for instance. And this coding RNA can adopt a small three-dimensional modifications that actually make it control its own expression. But we also have non-coding RNA, and we can divide this non-coding RNA broadly in a small non-coding RNA, like what we call the microRNAs of the PIWI systems, that actually play a key role in regulation of biological systems. Or we can have the large non-coding RNA, like the ribosomal RNA, the transfer RNA, the long non-coding RNAs that we know now play an important role as an antiviral mechanisms in some cases. If we want to have DNA, we need, of course, the exoribonucleotides, and in all living entities, the oxyribonucleotides are formed from ribonucleotides. And in my own lab, we have been concentrated on the enzymes involved in the modifications of ribonucleotides, like coenzymes, like histidine, which is a key amino acid in biological catalysis, like alarmones, which are actually signal molecules made of RNA that what we consider secondary messengers in biological systems. And we can have also the modification of some ribonucleotides to make antibiotics. In other words, RNA plays a key role in genetic store as a genetic polymer, in the synthesis of amino acids, in regulation, in antiviral defense, as precursors of DNA, and of course, in a number of small molecules. So what is the RNA world? Well, in my lab, we believe that, we believe that there are many, many definitions of the RNA world, including several that are contradictory, but I think that it's feasible to say that one can say that it was an early, perhaps primordial stage in which RNA molecules play a much more conspicuous role in heredity and metabolism. And of course, given the key role that I have already mentioned of ribonucleotide and modified ribonucleotides, I think they were also components of the RNA world as such. Now, if RNA was so important perhaps in the origin of life, but clearly in early evolution of life, how did it came into being? And here we have a tremendous debate, which has led to very, very active chemical research. Some people believe that the RNA world was the outcome of unexplored prebiotic chemical processes, that there are a number of chemical reactions that we have not explored in full that can give you functional RNA molecules, that can give you ribozymes that can act as replicases and so on. And others believe that no, perhaps RNA itself is the evolutionary outcome of other genetic polymers, what we call broadly the pre-RNA world possibility. And this is a very active field of research. But there's one point in which I think many, many of us agree, and it's the fact that you have RNA molecules that can replicate, RNA molecules that have ribozymic activity, RNA molecules that are key in many biochemical reactions. That doesn't mean that we have to adopt a very highly reductionist attitude, assuming that our life has started with a single molecule as such. The work of Pierluigi, formerly at the ETH in Zurich, the work of Dave Dimmer, the work of Jack Shostak and his group of Irene Shen in UCLA has demonstrated that, in fact, if you put RNA molecules and many other components between the boundaries of liposome, you actually have microreactors, you gain stability, you can open the possibility for additional reactions and so on. So the recognition of the RNA, of the significance of RNA molecules does not mean by itself that one should believe in a very, very simplistic idea of one living molecule emerging suddenly in the primitive Earth, but probably we have to think of systems of chemical molecules, systems of reactions taking place. So what happens with the original proposals of the heterotrophic hypothesis of the origins of life? Well, I think that we have modified quite strongly the ideas of Oparin to say that indeed we have many evidences that support the idea of there was a synthesis and accumulation of organic compounds in the primitive Earth, some of them brought in by comets, meteorites, asteroids, and so on, colliding against the primitive Earth. So we have good evidence for the soup. I think that we have good evidence for the very early role of key presence of RNA in early biological systems, but we may be wrong in extrapolating what we see today into the primitive soup. We really do not know how to get from the primitive soup into the RNA world. It's an open question. And needless to say, we have some insight, but many open questions about the transitions from these cells in which RNA play a key role into cells like the ones we see today with proteins and DNA as genetic material. Now, I was absolutely delighted with the fact that I was invited to speak in a series today that is devoted to the issue, as Tillman just mentioned, in the habitability from galactic to microbial scales. And believe me, when I saw the title of the series, I could not help but to think in an extraordinary painter, an extraordinary artist, Leonora Carrington, an English painter artist that went to live into Mexico, that say that the task of the right eye is to peer into the telescope while the left eye peers into the microscope. I think that is precisely what we have to keep in mind when we speak about the origins of life, and much more when we think about habitability from the galactic into the microbial scale. Thank you so much.
Thank you very much, Antonio, for this fascinating talk. We have already a question in the chat, but let me just first say that there are two ways of putting questions forward to Antonio. One is to write in the chat, in which case I will read the question to Antonio and the other participants, or you could raise your hand using the feature on the Zoom and then pose the question yourself. So the first question is by Hans Sineker. And he's asking, is it true that DNA and RNA have different 4 bases? Would this be important for the origin of life?
Well, that's a very important question, but in fact, they have more than 4 bases. Normally, we don't speak about the modified bases that we see, especially in RNA. And there is a recent fascinating paper, 2 years ago, a recent paper published by a French group demonstrating that you can have 2 aminopurine as a base present in viruses. There's a couple of very complete papers published on this issue in Science or Nature. And I have to say that the two aminopurine can be synthesized prebiotically. We reported one of those syntheses. I think it has, no, for sure, it has been found in meteorites. And there are recent papers by Jim Cleaves, also working on the origins of life, who has demonstrated that you could have manifold additional bases, and this could raise, in fact, the catalytic activities of genetic polymers. But there are the beautiful, beautiful work of Professor Albert Eschenmoser, a Swiss scientist, a universal scientist, I should say, that was in the ETH, demonstrating why the best possible combination in thermodynamic and chemical terms are precisely the basis that we see today. In other words, the components of RNA and DNA themselves have a very long evolutionary history behind them.
Okay, I'll read two more questions before we take the first, of those that had raised their hands. The William Ball asks, is there evidence of such RNA worlds in primitive areas of the Earth, like near active volcanoes or seismic faults?
No, the answer is no for today. In the prebiotic environment, I will imagine that RNA could accumulate either inside cells, if it's an invention of biological evolution, or if it could form prebiotically in places that are less active geologically, because it could destroy the molecule very rapidly.
Okay, Keith Horn asks, could the first life occur on wet dust grains or comets or asteroids in the proto-solar disk prior to the accretion onto the Earth?
I think it's very unlikely. I think that the experiments demonstrate that you require a wide range of environments, geological environments, big enough as to have different components, different interactions, and this will exclude the possibility of dust grains. The idea of cometary nuclei as hosting life was actually proposed years and years ago, but one could say that comets, because of their chemistry, their geology, are unlikely to host life. Some people think that perhaps in big asteroids like Ceres and Vesta and others, perhaps Vespa is a motorcycle in Italy, like Vesta, perhaps they had a geological history that could lead, if not to life, clearly to organic compounds. We see them.
Okay, let's take the first question from those who have raised their hands. So Hans Ludwig, Willy, could you make Hans Ludwig available? And Hans, could you open your mic and speak?
Can you hear me?
Vaguely, but maybe you speak up a little bit.
Okay, I get closer. Is that better?
That is better, yes.
Okay. Thanks, Antonio, for this fascinating tour through the origin of life. During the first part, so to say, of your talk, you showed the similarity between the chemical makeup of the Murchison meteorite and the Miller-Urey experiment, as far as I understood, right?
Right.
I was really surprised to see the similarity, since the conditions under which the chemical compounds are formed must have been very different. While the meteorite was chemical compounds were formed in the interstellar medium, the other was formed in this reducing atmosphere constructed in the laboratory. Is one in the first place has to expect that there's a similarity?
It's a very good point, Hans, and my answer would be that one should think of meteorites not as small pieces. Some of them are small pieces, actually, circling in the asteroid belt, but as component of larger protoplanetary bodies that then broke up. We have evidence, of course, in asteroids colliding among them, and the pieces are sent in different directions. They can eventually, some of them can come to the Earth. We can survive. We can analyze them, but don't think of the Murchison meteorite of a small piece in which you have all these chemical reactions taking place, but rather as part of a larger system. And one thing that I found particularly fascinating is the fact that you see a certain and I will put this in quotation marks, a certain level of chemical determinism, because you see the same synthesis, the same organic compounds forming over and over again. Basically amino acid, basically bases, and so on. But as the work that I show, the compilation of amino acids in different meteorites, the range of organic synthesis that was explored in the parent bodies of meteorites and clearly in the primitive Earth was huge. Let me give you an example that I have always found fascinating. We reanalyzed one of the samples that Stanley Miller left and found, for instance, norvalin. Norvalin is a 5-carbon amino acid that we don't see in living systems today. And it was actually formed much more abundantly in the experiment, in the analysis that we made. We demonstrated this was much more abundant than isovalene and valine. Now, valine is an amino acid that we see in proteins, we see in biological system. Isovalene and norvalene we don't see. So this is clearly telling you that what determined the formation of life was not only the not only the abundance of anorganic compounds, but a number of additional properties that play a role in selecting some of them over the other ones. And I think this is exactly what one has to think about what may have taken place in the primitive Earth.
Thank you.
Okay, let's move on to F. Diego. Diego, could you speak?
Yes, of course. Can you hear me?
Yes, we can hear you.
Hola, Antonio. Es Paco Diego. ¿Cómo está?
¿Qué tal, Paco?
My question is, we are talking about molecules. We are talking about something which is what makes life. It is not life itself. We're talking not talking about archaea or bacteria or anything like that. We are talking about our previous steps. That's one question. And the other one is there life which is not carbon-based? Because I get all these questions when I go to my talks, and then I say, well, all this is carbon, but there's a carbon, that's the life we know. Is there a life that we don't know, which is not carbon? And I say, well, no, it has to be carbon because of the amount of molecules that we get in space from carbon and the richness of that. Thank you.
I think you're quite correct, Paco. It's a pleasure to say hello to you. I suppose you are in London enjoying a wonderful cup of tea.
That's correct.
And let me first of all say, you are absolutely correct in terms of what we can synthesize, not always so easily, but we can synthesize a number of organic compounds of monomers. We have a little bit more problem with polymers, but we can see, in oligomers, but we can synthesize them. And one should not confuse the presence of organic compounds with the presence of life. As my friends who are mathematicians like to say, organic compounds appear to be, if I can rephrase their words, appear to be a necessary but not sufficient condition for life to appear. In other words, we should not be over-enthusiastic about the presence of organic molecules in other parts of the solar system, assuming that indicates the presence of life. No, not necessarily at all. Now, I think that the experiments with liposomes and systems of molecules are a huge step in demonstrating that we can go beyond the monomers into something that resembles some of the properties that we see in living cells. But we are far, far away from demonstrating that we can synthesize life on the left. On the other hand, it's true that that is not our goal in origins of life. What evolution, what we are demonstrating is that the origin of life was a stepwise process, perhaps not so slow, and that some of the steps we can readily simulate on the primitive, we can readily synthesize simulating the primitive conditions. I think that I have only spoken today about prebiotic synthesis, analysis of meteorites, and so on. But one also has to keep in mind that we can use bioinformatics to go back much back and forth that we see in living systems today. And that complements but trying to reach the issue of the origins of life by going from what we call the bottom-up approach to the upper to the lower approach using bioinformatics. But you are quite right. We have not been able to synthesize life, but that is not our goal, I have to say. On the other hand, the question that you raised about carbon-based life, it's the only kind of life that we have, that we know of. Carl Sagan used this very catchy phrase that we may be carbon chauvinist, but there is a fascinating book that I strongly recommend to you all, a book that was published in 1919 by Henderson, an extraordinary physiologist and chemist that was working in Harvard. And the book is called The Fitness of the Environment. And there Henderson analyzes the atomic composition of life. And he shows that all the organic compounds that we have, all the organic compounds that we see, and of course we know much more than he could imagine, but all life is made of the simplest and most abundant atoms in the periodic table. Carbon, hydrogen, nitrogen so are the simplest and most abundant atoms in the universe. One could always have a skeptical student, and I love them, I love them, but that could say, okay, but what if we have an unknown element that lies between 5 and 6 protons in the periodic table? And the answer right away comes, okay, you can't, because you do not have a number that comes, how do you call them? An integer that comes between number 5 and number 6. You can either have an atom with 5 protons or an atom with 6 protons, but not one that have 5.7, let us say. So if we look at the periodic table, that's highly deterministic. What we really see is that life is made of the most abundant and most simple atoms in the universe. Now you could say, what if there are different things? Well, let me quote Shakespeare, Hamlet. Horatio, there are more things in heaven and on earth than you can imagine. So perhaps we will see some surprises in the future, but not at the time being.
But what about silicon? Silicon is often quoted.
Yes, silicon is fascinating, and that has been a detailed exploration of its possibility. But don't forget that a living system is an open system, thermodynamically exchanging energy and material with its environment. And for instance, a key component in biological cycles on Earth is CO2, which is a gas. SiO2, the silicon equivalent, is wonderful for making glasses, but not good as a gas that you can actually use for exchanges.
Okay, let's get back to there are so many questions in the chat. Luca Tognetti asks, thank you very much, and many people say thank you, but I can't read them all to you. But what do you think about the prebiotic template-directed peptide synthesis based on amitoids by Greenwald et al.? Can short peptides direct the sequence-selective, regio-selective, and stereo-selective condensation of amino acids?
That's a fascinating question, and I would say it's an open question. Once you have an stereoselective synthesis, you could amplify it, and there are a number of beautiful essays demonstrating how this could take place, in which chirality is selected as a secondary possibility, and not as because it has a selective value on itself. There are a number of fascinating results and fascinating proposals. A recent one from Nice was published a couple of days ago by Ute, Cornelia, a number of friends I have down there. And indeed, it remains an open question. But I will say that whatever proposal we have, it has to realize that the physical and chemical properties of both L and D amino acids or L and D sugars are identical. So clearly the selection goes beyond the level of the monomer, goes into the level of the oligomers and polymers, and that remains an open question. But yes, I think it's a very interesting possibility.
Okay, Jérôme Benavist asks, all the chemistry bricks are there at hand for sourcing life. What is the prospect that low-frequency modulated electromagnetic waves amplified by our planet's ionosphere were the orchestra conductors, to organize life, RNA, DNA, protein synthesis?
Like you, I like symphonic music, but I think that the magnetic interactions will not probably lead to the level of such selection at the level of the surface of the Earth.
Okay. Now, Nico, I can't read your question of what it is because I can't read the signs that are in there. But TSVI, do you expect, though, that all sort of life in the universe will be based on the same basic molecules? I think you answered that question already. Is there a change, a different kind of chemistry for life? I think that has been addressed by you already. Or do you want to add a comment to it?
No. Sorry, Tilman. Yes, I missed that briefly. I think that one should be open for surprises, but my surprise will be tempered by the fact that you could have slightly different components, but not a completely different possibility.
Okay. Alan Sasha Brun asks, have you new updates on the chirality problem? The fact that the first sugar are right-handed and DNA left-handed? Where is the hilarity coming from? Interstellar medium and stellar radiation mediated? Chirality coming from?
Clearly, there's a mistake about chirality, not hilarity. But sometimes our explanation could be hilarious. So let me profit from that. I think that, again, if you go at the monomer level, you see the essentially same properties of L sugars and D sugars. But if you go at the next level of selection, there the question is different. I don't think that the chirality originated from the interstellar medium, but one of the first proposals for the selection of L amino acids over D amino acids was made precisely thinking of supernova interaction with beta particles and supernova explosions and so on.
Sander reflects on the telescope and microscope quote that you had. But then the question is, in regards to RNA, do you think life, if there is extraterrestrial life, it will have the same RNA basis that we have?
And no, not necessarily. The range of bases that you can incorporate is huge. I think that we have a strong reasons for understanding the presence of the bases that we see in RNA and DNA today, but that may not necessarily hold in other places. But remember that this is a quotation I often use. Many people, extraterrestrial life is like democracy. Everybody speaks about it, nobody has seen it.
Steve asks, the gap between RNA and the complex world of DNA, RNA proteins seems so vast that I do not see what processes could have bridged it. Can you comment on this, please?
I am delighted that this issue has been raised. I completely agree. I agree that the gap is huge, In fact, since Paco Diego is in London, let me remind you that when you get off the tube, the metro in London, the conductor always says, mind the gap, which is exactly what we have to do in Origins of Life. We have to mind the gap between these different stages. We have a good understanding, reasonably good understanding of the prebiotic soup. We have a huge gap in going from the prebiotic soup into RNA. We have a good understanding of RNA. We have of the possible RNA world. We have a huge gap in going, like Steve says, from RNA into the extant cells. But we have tools. For instance, we is not the royal we, but my lab and myself, we proposed about 5 years ago that the ubiquity of monomeric RNA polymerase, in other words, the RNA, the molecule, the protein molecule that replicates RNA viruses that is homologous to some polymerases that we see on cells, is actually an indication of what may have been the primitive RNA polymerase. You can use You can use bioinformatics, you can use three-dimensional comparisons, you can use sequence comparisons, and you can go back a little bit further back in time. Let me put you another example. ATPases are extremely complex molecules, one of the most complex molecules that we see in extant living entities. And it has, there's a part, the hydrophilic part of the ATPase has 3 alpha subunits and 3 beta subunits, and they are homologous. So clearly, they must descend from a molecules that had an ancestor to alpha and beta. So then you can picture a simpler ATPase. If you look at the elongation factors that are involved in protein biosynthesis, you see that there are 2 or a number of them, but let us concentrate on the two main ones. They are clearly homologous. So there must have been a time in which you only had one ancestral type for both of them. And in that way, you can go a little bit further back in time, and so on.
Okay, I have, I think, 3 more questions. There was one. What do you think about short RNA or short tRNA in cell communication during evolution?
Oh, that has been proposed indeed. Actually, there are a number of proposals that you could think, for instance, of a transfer RNA that was made by smaller RNAs that came together, and then a whole unit was selected, a whole big, well, it's not so big, 120 bases or so, was selected because it was more easily regulated in that way. Yes, it's a possibility.
Okay. Paulo Bolaro asked, what about chirality? Could chirality be used to discriminate extraterrestrial from terrestrial life?
At least we can use it to discriminate between the organic compounds that come unbound in meteorites and those that we form on the Earth. Now, don't forget that given enough time, a chiral a non-chiral mixture will become chiral. Don't forget that the enantiomeric properties change in time, and these are dependent of temperature and so on. So that is something that has to happen.
Okay, the question by Katja Nunez is there an environment on Earth that could serve as a model to study theories of the RNA world?
Not really, because remember the famous letter that Darwin wrote to one of his colleagues. If those compounds were synthesized today, they will be readily eaten up by the extraordinary amount of bacteria and other microbes that we see today. Microbes will chop up everything that you see around, every organic compound. Everything is potential food for them. So we don't have a primordial system on Earth that we can go on and say, here is where life may appear or is appearing. We see some more, we see some systems, some environments that we can picture as existing on the primitive Earth, but not necessarily an example of, this doesn't mean that life is developing there right now. But we have labs, so we can do it in the lab.
Okay, I think that uses all the questions that we have in the chat. So thank you very, very much again, Antonio, for this excellent start off of our series, and thank you very much for joining us. We had about 300 people listening in, which is an excellent start with the series, and I'm looking forward to meeting you all again. The next talk will be in 2 weeks' time, and my colleague Maurizio will be hosting it. But then a few talks later, I will be back and hosting the talks. So thank you very, very much, all, Antonio, and those who are listening in, and see you again. Bye-bye.
Bye-bye. Thank you.
Thank you.